Hi, I'm Barbara Imperiali. I'm a professor of chemistry and a professor of biology here at MIT. And I've been here since 1999 in my current disguise. I was actually a graduate student here many years ago. So a little bit about where I came from. I grew up in a village in England, and actually until I was 11, I was in a one-room school where there were 4 different grades all crammed into one room. And then at the end of that, I really was very lucky to be able to go to the British grammar school system, where in England you can really specialize early in certain subjects. And while I don't think I was very good at a lot of things, I just loved science. And so the British school system was great for me because I could specialize in chemistry, physics, biology, and math right on from the age of 14. The other aspect of things that made me really be able to move forward in the field was that the school I was at, there were very, very few people actually were interested in science. So most of my classes, there were only 4 of us in them. And so we really got special attention going through grammar school. Then I moved on to do my first degree in medicinal chemistry, which was where I started to bring together my interests in both chemistry and biology, and really starting to understand how molecules in life work and how we might be able to design small molecules to alter the action of certain enzymes that might be useful for treating disease. So I've really had an interest in this interface between biology and chemistry almost since the very beginning. I'm fascinated by the molecules of life and how we can understand them at the really detailed level. After that, I finished my undergraduate degree and shocked everyone at home by deciding to come to the States to do graduate work. And I came to MIT first in 1979 and did a degree in synthetic organic chemistry, which really was something that I did not necessarily thinking long term that was going to be my focus, but I wanted to learn how to build molecules so that I could probe biological systems. And I think that was a great thing to be able to do because it gives me a special talent in the way I attack biological problems. So after that degree then, my graduate degree, then I really started to move into the field of biochemistry, doing postdocs first at MIT and then Brandeis University in the field of biochemistry and enzymology. And enzymology is an interesting topic. We don't really call it that anymore. It sounds like an old-fashioned term, but really is about the mechanics of how enzymes work and catalyze biological reactions. So it really is a field that I was drawn to because I liked the organic side of things, but I also liked the functional side of the biological side of enzymes. By the time I was finishing my postdoctoral work, I really realized I was pretty unlikely to go back to England because I saw amazing opportunities in the United States to be able to start my own independent career in these fields of biochemistry and biology. There just seems to be so many more opportunities here than I thought I would have back in England, and so was very excited when I got the opportunity to move to Caltech to start my own independent career, running a research group, teaching, and really bringing together these fields that I'd spent a long time studying. So in my independent career, I spent about 10 years at Caltech and then had a very exciting chance to return to MIT. When I came back here, I really felt like I was coming back home to Boston because that was where I'd spent so many happy years as a graduate student. And I first came back, I moved into the chemistry department and have more recently moved over into the biology department here, where I think it's a great environment because I feel like my chemistry and biochemistry roots are strong, but it's exciting to be able to interact with the community here to understand ways in which these skills that I've developed can be applied to probe biological systems. So my current research would be defined as chemical biology. The best way to describe that field is the application of chemical and physical approaches to study complex biological systems and really dig deep into understanding the molecular basis for biological function. So in my research, I work in the field of glycobiology, which is just a fancy word for understanding the biochemistry and biology of sugars or carbohydrates, as they are often termed. And I think in the class in 701, you've seen how sugars are very, very important and central in metabolism, but there's a whole other role that sugars have in biological systems, and that's when they are exploited to decorate protein structures. And I know that might sound like a little bit like a birthday cake when we talk about sugars decorating other things. And in a way, sugars really do add the decoration to proteins that can be very important for their function. In biology, you've learned about a post-translational modification called phosphorylation that's important in signaling. Glycosylation, the addition of sugars, is important for other aspects of biological function. And I actually made a few images here because I wanted to show you when we think about protein structures, and I've shown you here a beautiful sort of ribbon diagram of a protein structure. But what I wanted to show you is how sugar groups can be very large and can really alter the way a protein might look. So when you first look at this sort of cobweb of atoms and molecules, things don't stand out very well. But if I highlight where the sugars are, I can show those sugars as spheres, so just to give you a feel for their size altogether. And then I can basically take a look at the rest of the protein structure and put like a coat on it, a mesh structure over it. And you see that the protein might look like a large molecule, but the sugar also has a great impact on the surface of that protein. But you can see how it alters the landscape of the protein. And so sugars on proteins in biology often play very important roles in how those proteins interact in biological systems. And very often proteins that are on the surface of the cells become glycosylated with these kinds of structures, which impacts how cells communicate with each other. And there's a very serious issues or serious health disorders will occur when the machinery that's responsible for adding the sugar or building the sugar up to this complex structure isn't correctly formed, and there can be serious, very serious developmental consequences. And so we're really intrigued by the possibility of understanding not just how sugars affect proteins, but the pathways that build up those sugars and add them onto proteins. So in the past few years, my group has turned its attention to understanding the sugars that are attached to bacterial proteins. And in particular, there seems to be lots and lots of new information coming out that many bacterial cell surface proteins become glycosylated. And while sugars are important on human proteins for things like development and cell-cell communication and the establishment of tissues and organs, in bacteria they're there for much more sinister reasons. And in fact, it seems that sugars may decorate bacterial proteins in order to make bacteria be able to avoid the immune system. This has been a topic you've talked about lately in biology. So sugars kind of mask that bacterial protein and let the bacteria kind of sneak into the physiology of human systems. And in particular, glycoproteins have been associated with pathogens. And these are the kinds of bacteria that will do harm to you and cause disease. Another place where bacterial glycoproteins seem to be very important is when they are displayed on the surface of bacteria. They affect how the bacteria interact with human cells and form, in a sense, they're part of the glue that makes the bacteria stick to the human cells, enable them to adhere to and invade human tissues, and in fact cause more harm. So what we've been trying to do over recent years is start to think about whether the pathways that lead to bacterial glycoproteins could possibly be a target for intervention in disease and infectious diseases, where, as you probably know, there's a lot of problems right now with humans gaining resistance to common antibiotics. And our community is thinking very hard about how we might be able to come up with strategies to get around antibiotic resistance. So what my group is doing is trying to target some of the enzymes that are involved in the assembly of bacterial glycoproteins. So here, once again, I've tried to show you, this is a protein that's a target that ends up being an enzyme that's involved in glycoprotein biosynthesis in bacteria. I've defined it with a ribbon diagram, but I think these things are really most cool if you start moving the ribbon around. You can see this is a really interesting structure. You've talked about beta helices, alpha helices and beta sheets in class. There's a very unusual fold in this protein. It's actually called a beta helix. It's put together by strands of beta structure forming this coil. You can almost look down it. So this is the structure of an enzyme. We've solved the structure of this using X-ray crystallography, and we're trying to design inhibitors that will stop the normal substrates binding to this enzyme. A normal substrate is shown here. This is a structure known as acetyl-CoA. I'm just going to show you that structure. There it is, bound to the enzyme. You can't quite see the binding site, but if you coat the enzyme with a surface, you can start to see how that structure binds there in a very special pocket. Once again, I'm just going to stop this structure from clipping, but you see the spheres are the usual structure of a substrate that binds to this enzyme and acts on it and starts to build building blocks that go into bacterial glycoproteins. So what my group does is it takes a particular approach to finding organic molecules that can maybe compete with that structure and maybe stop its activity in order that we ultimately stop the assembly of bacterial glycoproteins. And here I've chopped forward to show you, in fact, I think these structures on screens are just so much more exciting. This molecule I have in my hand, we've actually solved its structure bound to that same enzyme, and you can see it sort of lying there instead of the usual substrate, taking the place of the substrate and stopping it from binding so there can't be any reaction. And as with the skills of organic chemistry, we can take these structures and also take the structure that's bound to the enzyme and say, how can we make this structure bigger so that it binds more tightly to the protein and becomes even more powerful at inhibiting the activity? You can actually look at the structure and you can say, okay, I see the structure here. Believe it or not, those two red oxygens are this molecule. The yellow is the sulfur shown there. And we can look at the big protein structure and then say, let's try and build our molecule into little cavities where there's some space there, and fill that cavity up with with a molecule that's been grown into that space, make something that binds even more tightly, that might be more potent to inhibit the enzyme. And if these molecules inhibit the enzyme, then we can start to do tests in biological systems, either in the bacteria or in animal systems, to test whether we stop making glycoproteins, and in fact whether these bacteria become less pathogenic or less harmful when we do that. So the other part of me at MIT is teaching, and I will tell you that I love to teach. This is really, I get so much energy from it. I guess I think I spend an enormous amount of time getting ready for classes, but it all comes back to me when I get in the classroom and I find ways to connect with the students to explain this border between chemistry and biology and the molecular basis for biology. And so I think it's a really exciting opportunity to be able to share with people structures, how we can, as chemical biologists, inhibit activities and potentially start to contribute towards making molecules that might one day be relevant in treating disease. MIT is a really exciting place to be able to teach because I think the students are hungry for the kinds of things that we're doing here in the biology department. Biology interfaces with so many disciplines at MIT, with chemistry, as I've already described, but with engineering, with computation. And I think that the students who are drawn to those interface types of disciplines really get a lot out of MIT because they have the opportunities to come into the lab and really learn not just one focused discipline, but learn how to synthesize all these disciplines together in the pursuit of new investigations. In the fall, I'm pretty excited. I've taught organic chemistry for a long time, but in the fall I'm going to be teaming up with some of my colleagues to teach general biology, and I hope to bring into the course some of the principles I've been describing to you today. And I think that, that it should be something that will capture the attention of students, because I think our current generations are very interested in really delving deep in how we can contribute at the most fundamental level to understanding the challenges of biology at a very fine detail of the molecular detail. I run a smallish research group. It's about 12 students, 6 postdocs and 6 graduate students, and they come from all over the world. And I think what's really excellent about having the opportunity to run a research group here is the interactions amongst the team and how they synergize so well with each other. I always feel that our research groups are so much more than the sum of the parts, and the opportunities to get into the lab and ask people about what they're thinking about with respect to particular problems, and sit down with them at the computer and start designing new molecules and ways to synthesize them is really very exciting, and it brings me in every day excited to face these new opportunities. My colleagues in biology are really a fantastic crowd. It's no problem to shoot someone an email and say, I'm not sure I'm thinking about this right, can you help me out? And you almost always get a reply email within an hour saying, stop by and let's chat about these problems. So that's also, we have a very interactive department that I think allows us to solve big problems in different ways. I'm really grateful to have had the opportunity to talk to you about my research, and thank you for listening. And I hope one day you get the opportunity to share some of these exciting research opportunities at this very fascinating border of biology with chemistry and biochemistry.